flashback what we have discussed on previous class earlier. So if you still remember, on our last class last week, we discussed about the potentiometric methods. Okay, so basically this method use the chemical reactions in a solutions. Okay, specifically we use redox reactions. So what is redox reactions? Redox reactions is a chemical reactions where one elements will be reduced and another elements will be oxidized. So redox reactions occur simultaneously. Okay, so these both reactions occur at the same times. Okay, so once an elements become reduced, another elements will be oxidized. Okay, because in these chemical reactions they are the electron transfer. Okay, so what is reactions? Reduction reaction is when an element receive an electrons, okay, lost oxygens, okay, ataupun increase its oxidation number. Whereas oxidations, what is oxidations? When an element lost electrons, okay, or receive oxygens or reduce in its oxidations number. Okay, so apart from that, another important things in redox reactions is redox reactions can generate electrics. Okay, betul tak? Apa dia cells yang kalau kita gunakan untuk generate electrics, apa nama dia? Siren. Mana Siren dah hilang dah?
Tak ada. Saya electrolyte tu kan?
No, apa nama cells yang kita panggil untuk generate electric tu?
Galvanic cell ke doktor?
Yes, galvanic cells. Okay. Okay. So that's the important point that you need to understand about the potentiometric methods. Okay, you must understand what is redox reactions, what is the uh, main functions of redox reaction. Actually, redox reactions you want to create the electricity. Okay, and then I want you to understand the components in galvanic cells. We have electrodes, we have sub ions of the electrode elements. Okay, we have junctions, and then we have a device to determine or to measure the electric currents. Okay, so that's the important point or the important topic that you need to understand and also remember when we discuss about the redox reactions and also potentiometric methods. Okay, so let us continue with our new topics, which is the potentiometric methods. Okay, so what is potentiometric methods? So you must remember, in order for redox reactions to occur, we must have the potentials. Okay, what is by the potentials? What is electrochemical potentials? Electrochemical potentials is when we have two elements, okay, or two ions. Okay, so these two ions, ones have the ability to reduce and one have the ability to undergo redox reactions. Okay, so if we have two elements where both elements have less potentials, electrochemical potentials, the redox reactions Okay, class. Can you hear my voice? Jelas tak suara saya ataupun too distance?
Jelas.
Okay, sorry ya. Wireless problems. So saya tanya Dr. Fahmi dekat sebelah, memang wireless tengah problem. So sorry, tiba-tiba saya terkeluar. So wait, saya nak share balik skrin saya. Saya tak ingat, boleh nampak tak skrin saya?
Boleh.
Okay, sorry. Inilah yang saya tak suka bila buat kelas online. So biasa tak lancar. Okay. So saya tak ingat saya berhenti dekat mana, tapi saya nak all of you understand and remember what is redox reactions, okay? And also remember the components of galvanic cells, and then you must understand the electrochemical potentials, okay? So these topics you need to understand for you to much understand about the potentiometric methods, okay? So basically what is potentiometric methods? These methods measure the electrical current generated by the ions in a solutions, okay? So basically we use potentiometric methods to measure the concentrations of ions, okay? So remember, if the concentrations of ion in a solution is higher, so it will create or generate higher electrical currents. So that's what we measure in potentiometric methods. So it depends on what ion we want to measure, okay? Example of ion that we can measure using potentiometric methods are pH, okay, the hydrogen ions, or the ion in soil solutions, okay? So remember, to determine whether the soil is fertile, subur atau tak subur adalah kita ukur kandungan the available nutrients within the soils, okay? And in order for the nutrients to be available for the plant uptake, it must be dissolved in soil solutions, or in other words, it must be exist in ions, okay? So therefore we can measure the concentrations of the ion in soil solutions using the potentiometric methods, okay? So this is the basic components in potentiometer, okay? Regardless whether the instruments used to measure the pH concentrations, the ion concentrations, ataupun kita nak measure apa-apa ions, dia ada 3 components ni, okay? The first one is reference electrodes, okay? So what is reference electrodes? This electrode will give reference for potential measurements, okay? And then we have the indicator electrodes where species of interest is measured, okay? And then we have potential measuring devices, okay? So biasanya ini adalah voltage meter, okay? Tapi kita akan ubah dia. Sebagai contoh, in pH meter we convert the electrical currents, the voltage meter into the pH meter, okay? So what is reference electrodes? So the functions of electrode is to act as a reference against the potentials measurements can be made, okay? So for example, in pH meter, we have a silver chloride as a reference electrode, okay? So the reference electrode must have a certain characteristic, a desired characteristic, okay, in order for these elements to be used as a electron reference, okay, reference electrode. It must known or fixed potentials, okay? We know its potential. What is apa maksud potentially? The electrochemical potentials, okay? And then it has constant response, okay? What is mean by constant response? Okay, for example, we want to measure the pH meter. It must respond to hydrogen ion. It will not respond to other any ions, okay? And then it insensitive to compositions of solutions under study, okay? So it must very insensitive to the compositions, okay? So for example, we have a solutions, okay? A salt solution, for example. In salt solutions, there are difference of ions. So the reference electrode must be insensitive to that ions, okay? So kita ada satu lagi method measuring, kita panggil apa? Indicator method. Indicator electrode ni yang sensitive to that compositions of the solutions under the study. So the reference electrode must be insensitive to the solutions under the study. And then another important characteristic of reference electrode, it must obey the Nernst equations, okay? What is Nernst equations? Basically, Nernst equations calculate the electrochemical potentials of the elements, okay? So in general, Nernst equations explain that the higher concentrations of ions, it will generate higher electrics. Okay, so if a solutions contain higher ion solutions, we can generate higher electrical currents from that solutions. Okay, so Nernst equations relate the concentrations of of ion in solutions with electrical currents. Okay, it must reversibles. Okay, because why it must be reversible? Because we want to measure the ion in our samples or in the solutions many times. Okay, kita tak boleh lah guna sekali saja. Kalau it irreversibles, okay, kita hanya boleh guna sekali saja. Kita tak boleh gunakan banyak kali. Okay, so pH meter yang kita gunakan dekat lab kita boleh measure the pH many times because the reference electrode are reversibles. Okay, and then this is the common examples of reference electrode used in potentiometer. Okay, for example, we have column electrodes contains mercury chloride and also potassium chloride, and we have also silver or silver chloride electrodes. Okay, so basically in our pH meter we use silver chloride electrodes. Okay, then apart from reference electrodes, we have indicator electrodes. Okay, the main functions of indicator electrode is to detect or respond to the presence of analyte or ions in our samples. Okay, so this electrode is the one that measure the concentrations of ions in a samples. Okay, the three common types. Okay, we have a membrane indicator electrode or ion selective electrode. We have metal indicator electrode. We have first hand metal indicator electrodes. Okay, tapi in our class we will focus on ion selective electrodes. Okay, because this is the common ones. Okay, usually used in pH meter, EC meter, or elements meter. Okay, so what is ion selective? So nanti saya akan terangkan apa itu ion selective electrode. Okay, so indicator electrode determines the concentrations of ion based on selective absorptions of ion in the membrane surface. Okay, so this is how ion selective electrode determines the concentrations of ion in the solutions. Okay, so remember in our solutions, okay, for example, we want to determine the pH of solutions. Okay, we have, for example, we have sulfuric acids. Okay, so in the solutions we have sulfur ions, we have water ions, and we also have hydrogen ions. Okay, but the ion that we want to measure, the concentrations of ion that we want to measure is hydrogen ions. Okay, so the indicator electrode must be sensitive to hydrogen ion. It must detect, okay, or measure the concentration of hydrogen ions. Okay, so in order to determine or detect the concentrations of hydrogen ion, the ion selective electrode will select hydrogen ion only. Okay, sebab tu dinama dia selective electrode. Dia very selective. Dia hanya pilih ion tertentu saja. In this case, it will select the hydrogen ions. Okay. The general mechanisms by which an ion selective potential develop in this device depend on the nature of the membranes. The potential of metallic electrode arises from the tendency of oxidation reaction occur at an electrode surface. Okay. In membrane electrode, the observed potentials is a kind of junctions potentials that develop across a membrane that separate the analyte solutions from reference solutions. Okay. So this statement is very technical. So nanti saya akan terangkan satu-satu the statements. Okay. So in galvanic cells, if you still remember, we have salt bridge. Okay. What's the functions of salt bridge? Salt bridge used to connect between the two the solutions. Okay, so kalau awak ingat, in redox reactions kita boleh separate the cation electrons and ion electrons into separate solutions. Okay, however, in order for oxidation reactions to occur, these two solutions must be connected. And in galvanic cells or voltaic cells, the solutions are connected by the salt bridge. Okay, however, in pH meter, uh, the 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 circuits, okay, the solutions is connected by junctions potentials. Okay, so what is junctions potential? So remember, in our pH meter we have ion selective electrodes. So these ion selective electrodes select specific ions, okay, to pass to its membranes. Okay, in this case it only select hydrogen ions. Okay, so let imagine, so we have two solutions, and these solutions are separated by very selective membranes, okay, or porous membranes. Okay, so we have uh, two solutions. Okay, for example, in here we have um, chloride ions, and then in X solution and in Y solutions we have only water. Okay, so what happened? The chloride ions will diffuse into a lower concentrations. Okay, ini adalah simple diffusions. Kita ada satu high concentrations which is in X solutions, and then lagi satu kita ada low concentrations solutions which is Y solutions. So what happens bila kita biarkan dua solutions ni, lama-lama apa berlaku? Dia akan ada equilibriums. Okay, so the ions concentrations on both solutions will be in equilibriums kalau kita biarkan lama-lama. Okay, tapi untuk proses tu berlaku, ions tu dia tak terus equilibrium. Okay, dia akan ambil masa. Okay, for example, we have an ions and cations. Okay, for example, in here we have hydrogen ion and chloride ions. Hydrogen ion is smaller compared to chloride ions, so it will move from high concentration to lower concentrations faster compared to chloride ions. Okay, because of hydrogen ion is positive and chloride ion is negative. Okay, so before both those solutions reach equilibriums, the both solutions in the process they akan ada different electrical or charge gradients. Okay, so yang dekat sini dia akan yang Y ni dia akan ada excess of positive ions. Okay, sebab apa? Hydrogen ion ni dia lebih kecil, okay? And then it easily diffuse into Y solutions. Okay, so before the both solution in equilibriums, ini yang akan berlaku. Okay, sebelah X akan excess negative anions. Okay, sebab chloride ni dia lebih besar, dia lambat sikit untuk diffuse into Y solutions. And then the H ions, hydrogen ions, dia akan move faster. Okay, sebab dia lebih kecil. So apa yang berlaku? Dia ada different charge gradients. Okay. So these phenomena will result in electrical double layer of positive and negative charge at the junctions of two solutions. Okay, thus at the point of junctions, a potential difference will develop because the ionic transfer. Okay. So ini susah nak faham, nanti saya akan terangkan. Example of ion selective electrode is pH meter. Okay, so pH meter is common example of ion selective electrodes. So this indicator electrodes in pH meter, it is based on use of glass membranes that preferentially bind to hydrogen ions. Okay, so ini adalah contoh ion selective electrodes. What is ion selective electrode? It will select specific ions. Okay, so in this case, the pH meter will only select the hydrogen ion. Okay, so remember, in order to redox reactions or electrical currents to be generated, okay, the solutions must be connected. In galvanic cells, the two solutions are connected by salt bridge. Okay. In pH meter, the two solutions are connected by junctions potentials. Okay. Sebab apa berlaku junctions potentials? Sebab ada difference gradients of ions. Okay. The difference ion positif and ion negatif, dia akan membolehkan electrical current to pass through. Okay, dia akan jadi conductor dekat sini sebab ada different junction potentials. Okay, okay, this is the diagrams of the pH meter. Okay, we have two different electrodes. Okay, we have reference electrodes. Okay, made up of silver chloride, and then we have reference electrodes, mana satu ya? Indicator electrode. Indicator electrode tu adalah glass membranes. Okay, we have two electrode. Okay, ingat dalam potentiometric methods kita ada dua electrode. Satu adalah reference electrode. Okay, so lagi satu electrode dekat sini adalah ion selective electrode. Okay, in pH meter we use glass membranes. Okay, tapi glass membrane ni bukan sebarangan glass. Okay, so glass membrane ni dia hanya very selective to hydrogen ions. Okay, and then we have a solutions. Okay, reference solutions. In here reference solutions kita gunakan potassium chloride. Okay, potassium chloride pH dia adalah pH 7. Okay, and then we have another solutions. Okay, biasanya another solutions ni adalah salt solutions of silver chloride. Untuk silver chloride ni tadi. Okay, so we have salt solutions for silver chloride in order for redox reactions to occur. Okay, so in glass pH probe, it contains two electrode, a sensor electrode and a reference electrode. Okay, these electrodes are in the form of glass tubes, contains pH buffer, okay, pH 7 buffer, and other contains saturated potassium chloride solutions. The sensor electrode bulb is made of porous glass or permeable glass, membrane coated with silica and metal salts. A silver wire coated with silver chloride is immersed in pH 7 buffer in the bulb. Okay, another silver wire coated with silver chloride is immersed in the saturated potassium chloride solutions in reference electrodes. Okay, so whenever there is a charge imbalance across any materials, there will be electrical potential across the materials. Okay, so this is in redox solutions. Okay, the concentrations of proton inside the membrane is constant, and the concentrations outside is determined by the concentrations or the activity of the protons in analyte solutions. So the concentrations difference produce potential difference that we measure with a pH meter. At each membrane solvent interface, a small local potential develops due to preferential absorptions of hydrogen ion onto the glass surface. Okay, so susah nak faham, so saya terangkan. So we have a pH meter. Okay, so remember in pH meter we have the two electrode. One is reference electrode with each silver chloride metals, and then we have indicator electrode, which is a glass membrane electrodes. Okay, so ini adalah gambar kita punya indicator electrode. Okay, ini adalah a glass bulb tu tadi. Okay, so kalau awak nampak ini adalah warna kelabu dan biru ni, ini adalah kita punya membrane ni. Okay, glass membrane ni. Okay, so faham tak yang bulat ni? Ini adalah diagram dia. So ni adalah kita punya indicator electrode. Okay, so this indicator electrode is made out of glass membranes. So this glass membranes is very selective. It can only select hydrogen ion. Okay, so imagine we have kita ada apa ni potassium chloride for example. Okay, ataupun kita ada solutions. In these solutions, it contains chloride ions and also hydrogen ions. Okay, so hydrogen ion is smaller than chloride ions. So this is ion selective membranes will allow the hydrogen ion to pass through it membranes. So what happens? Okay, so ingat tadi dekat junctions potentials kita ada dua solutions with different concentrations. So apa yang berlaku? The high concentrations ions ni dia akan pergi ke dua-dua tempat, ke both solutions. Okay, tapi dia punya dynamics, dia punya laluan tu high concentrations lebih kepada lower concentrations. Okay, so bila kita immerse the indicator electrode in solutions, what will happen? The glass will allow hydrogen ion to pass through. Okay, so the hydrogen ion in reference solutions will do the same things. Okay, dia akan move to the membranes. Okay, and then what happens? If this happens, okay, apa yang berlaku? Dia ada junctions potentials. Okay, so this, because of these membranes contains hydrogen ion, dia boleh alirkan elektrik. Okay, dia jadi conductor. Okay, when it become a conductor, it can allow the electrical current to, okay, so if we have the same concentrations of hydrogen ions, okay, what will happen? The reference solutions, the amount or the concentrations of hydrogen ion that attach in the inner side of the ion selective membranes is equal to the solutions that we want to measure. Okay, so kita ada pH 7, bukan pH 7, for example kita ada acidic solutions. Okay, so apa dia acidic solutions? Acidic solutions contain high concentrations of hydrogen ion. Okay, betul tak? Kalau kita ada satu solutions yang pH sangat acidic ataupun pH dia rendah, the solutions contain very high of hydrogen ions. Okay, so what will happen if the hydrogen ion, okay, dekat luar ni tinggi, the electrical currents that can pass through in these membranes adalah lebih tinggi. Okay, so bila dia lebih tinggi, kita detect dia sebagai pH dia rendah. Okay, so kita akan hydrogen ion. Jika hydrogen ion dia tinggi, pH dia rendah. Okay, so in these situations, the hydrogen ion dia lebih banyak. Sebab apa? Larutan ni adalah acidic. So bila hydrogen ion lebih banyak, dia boleh alirkan elektrik dengan lebih baik. Okay, so kita akan detect the electrical current dia tinggi. Okay, so the pH meter dia akan convert. So, oleh sebab pH current dia tinggi, dia akan convert kepada pH yang nombor pH yang rendah. Sebab apa? Acidic. Okay, acidic, hydrogen ion dia tinggi. Okay, so what will happen if the concentrations of hydrogen ion in the solutions that you want to measure is equal to reference solutions? So the number of hydrogen ion, okay, in outer side of ion selective membranes and in inner side of ion selective membrane is equal. Okay, so the electrical currents that flow in these junctions potentials lebih rendah, okay, compared to solutions that contain higher hydrogen ions. Okay, so dia akan detect kalau electrical current dia hydrogen ion dia sama in both side, pH tu sama dengan 7. Okay, so what will happen when we have lower of hydrogen ion concentration? Okay, for example, in here we have alkaline ions, alkaline solutions. So in alkaline solutions, we have a very low concentrations of hydrogen ion compared to acidic solutions. Okay, so in here we have kita punya pH meter, kita ada reference solutions. Okay, and then we have solution yang kita nak detect tu, which is alkaline. So what happens? The hydrogen ion in ion selective membranes will be lower compared to in ion selective yang boleh lalu dekat sini in these junctions potentials lebih rendah. Okay, so bila electrical current lebih rendah, it will indicate the concentrations of hydrogen ion lebih rendah. So dia akan bagi bacaan pH lebih tinggi. Okay, faham tak? Abharina, faham tak? Okay ke tak? Abharina tak ada ke? Abharina, boleh dengar suara saya? So faham tak? Okay, kalau faham saya nak tanya. Kalau in galvanic cells, we have cell bridge. Tapi dalam potentiometric meter, we have components that function same as South Bridge. Apa dia? Abah Harina? Mana Abah Harina? Hilang dah Abah Harina. Iklimah? Yes. Okay. So in galvanic cells, we have South Bridge. Okay. So the functions of South Bridge is to connect between two solutions. So bila ada connections, barulah ada redox reactions. Okay. Tapi dalam pH meter, South Bridge tu tak ada. Kita kena develop. Okay. So macam mana kita nak develop? By the hydrogen ion. Okay. So if the concentrations of hydrogen ions dekat solution tu tinggi, which is acidic, dia dapat kita akan detect sebagai hydrogen ion dia lebih tinggi, electrical current dia lebih tinggi. So dia akan ubah daripada tinggi, solution ni adalah acidic, so pH dia rendah. Okay. So kalau alkaline, the hydrogen ion dia rendah, electrical current yang boleh lalu pun rendah. Okay. Sebab electrical current rendah, dia akan okay, so kita akan ubah electrical current tu kepada pH value, which is pH value dia tinggi. Okay. Faham tak? Iklimah, ada soalan? Iklimah, ada soalan tak? Faham?
Faham.
Okay, so Iklimah, kalau faham, boleh nampak slide saya tak?
Boleh doktor.
Okay, ini adalah galvanic cells. Okay, so galvanic cells ni jugalah yang kita gunakan as a principles to determine the concentrations of hydrogen ion ataupun kita nak tentukan the acidity, the pH of the solutions. Okay, so in these galvanic cells, we have two plates. We have copper plate and then we have zinc plate that serve as a electrode. We have anode and then we have cathode, positive cathode, and then we have negative electrode. We have solutions, okay, in which the redox reactions will occur in the solutions, okay, in both solutions. And then we have salt bridge. Salt bridge is to connect between the solutions. Okay, in galvanic cells, salt bridge contains ion. So that the redox reactions can happen. In pH meter, apa kalau dalam galvanic cells kita ada ions, okay, salt ion untuk dalam salt bridge ni, okay. Saya cakap bukan salt bridge ya, saya cakap berkenaan dengan ion dalam salt bridge. Okay. Dekat pH meter, apa ion yang memainkan fungsi yang sama dalam salt ion, salt ion dalam salt bridge ni? Faham tak soalan saya, Iklimah? In galvanic cells, we have salt bridge. Okay. In pH meter, we have junctions potentials. Okay. Both component play same functions. Okay. Tapi saya nak pecahkan lagi. Dalam salt bridge, kita ada salt ions. Okay. So, this salt ion yang akan menghubungkan, yang akan menyebabkan electrons boleh mengalir. Okay. Dalam pH meter, we have junctions potentials. Okay. Apa yang menyebabkan, ion apa yang akan menyebabkan ada junctions potentials tu? Iklimah. Mana Iklimah? Tengah berfikir ke? Faham tak Iklimah soalan saya? Mana Iklimah?
Hydrogen ion.
Ya, betul. Hydrogen ion. Okay. Sorry ya, saya tak boleh buka kamera saya. Kalau saya buka nanti dia akan lost connections. Saya nak tanya lagi soalan. Saya nak pastikan awak betul-betul faham. Saya tak ada gambar rajah yang lebih besar. Okay. Tapi saya gunakan yang ni lah. Okay. So this is sekejap ya. So ini last kali sebelum kita end our sessions today. So in here we have like a galvanic cells. Okay, so we have electrodes, which is and then we have reference electrodes, which is glass membranes, and then we have filling solutions, and then we have silver chloride, saturated calcium chloride solutions. Okay, so in order to determine the concentrations of hydrogen ions, okay, we must connect. Kalau pH meter ni kita tak rendam atau kita tak immerse dalam solutions, dia punya circuit litar ni dia tak sempurna lah. So electrical currents tak boleh berjalan. When we immerse the solution, the pH probe in a solutions, electrical currents boleh berjalan. Sebab apa? The circuits now connected. Okay. Sebab apa the circuit connected? Because the glass membranes dia boleh select the hydrogen ions. Okay. The strength of the electrical currents depend berapa banyak hydrogens yang boleh dia boleh apa ya? Dia boleh meresap dekat glass membranes ni. Kalau hydrogen ion semakin tinggi, the electrical currents will be higher. Kalau hydrogen ion rendah, electrical current become lower. The redox reactions, okay, happen between reference solutions and the buffer solutions. Okay, so redox reactions happen. The electrical current can be generated. Okay, so how much electrical current can be moved in these circuits depend on the hydrogen ions, the salt bridge, the junctions yang terbina melalui solution yang kita nak measure tadi. Okay, so any other questions? Ifah, okay tak Ifah? Mana Ifah ni? Jumpa tak?
Ada doktor.
Okay tak? Any questions tak?
Okay.
Okay. So apart from pH meter, apa lagi instruments yang gunakan potentiometric principle?
EC.
EC meter, yes. EC meter, okay. Apart from EC meter, okay, we have apa ya? Saya pun tak ingat lah. Biasa yang kita gunakan dua tu je lah. EC meter, pH meter, okay. Example in agriculture to analyze nitrate in soil samples, okay. Itu adalah EC meter. Okay. We have instruments that analyze very specific ions. Okay, for example, we have nitrates meter, okay, iron bromide, calciums, and etc. Okay, and in drinks, milk, meat, and fruits, we have biasanya kita ada specific potentiometric methods yang kita gunakan untuk detect ion dalam drinks, milk ataupun dalam fruit juice. Okay, so basically kalau fruit juice ataupun kita punya susu, kita punya meat tak basi, okay, masih elok lagi, biasanya ion-ion dalam solution tu dia tak tinggi sangat. Okay, kalau dia dah basi, dah rosak, biasanya ion lebih tinggi. Sebab apa? Decomposition process dah berlaku. Okay, apa dia decomposition process? Decomposition process is process to transform complex molecules into a smaller molecules. Okay, smaller molecules tu biasanya dalam bentuk ions lah. So bila kita detect concentrations ion tu lebih tinggi, so kita ada threshold lah. Okay, threshold, berapa threshold dia, concentration ion tu tinggi, baru kita detect dia sebagai data elok. Okay, so itu in beverages industry. Okay, apart from that, we have also instrument that analyze of chloride in paper paste, cyanide in electrolyte bath, chloride and fluoride in galvanic process. We also have especially in medical industry, we have a probe or instrument that analyze concentration of serums, okay, for example in our blood, okay, and also in biological fluids. Okay, dalam kita punya contoh kita buat blood test, okay, kita akan test serum je, berapa banyak proteins, okay, berapa banyak albumins, all those can be determined using potentiometric methods. Okay. So any questions? Any other questions before we end our class sessions? Syairin, okay tak Syairin? Syairin? Mana Syairin ni? Tak ada dekat sini ya, Syairin ya. Mas Rizal, okay Mas Rizal? Any questions?
Saya okey Doktor, tak ada soalan.
Okay. So thank you everyone. Okay, so this is the last sessions for my class next week. This afternoon. Okay, Dr. Fahmi told me that you have a sessions with him because we have a lot of holiday on Monday kan. Kita ada Christmas, kita ada Tahun Baru. Nasib baik hari ni tak cuti. So dah tahu ke siapa Perdana Menteri? Dah tahu dah? Tak tahu lagi ya? Okay.
Belum.
Belum ya?
Belum.
On week 8 kita akan ada lab sessions, okay, and then kita akan ada midterms, okay, midterms paling last minggu 10 tak silap saya, okay. So sebab hari tu Dr. Syahmi ada jumpa, dia akan buat field trips, and then dia punya field trips dengan awak adalah hari Isnin, so dia minta tukar. So nanti kita akan tengok lah lab dengan midterms tu akan buat hari apa, okay. So if there are no questions, so I would like to thank to all of you for your times, okay, for your concentrations. Okay, hopefully you are all understand what topic that being discussed in our class. Okay, so jumpa lagi dekat lab sessions kita. Okay, so assalamualaikum class, good afternoons, goodbye. Jumpa lagi.
Thank you, Doktor.
Attendance thank you, Doktor.
Thank you, Doktor.
Thank you, Doktor.
Saya dah print screen, saya dah tahu siapa yang ada, siapa yang tak ada.
Okay.
Okay, thank you.
Thank you, Doktor.
Assalamualaikum.